For centuries, blood types seemed like nothing more than random biological variation, scattered arbitrarily across humanity. Then scientists mapped blood type distribution across every continent on Earth. A genuinely striking pattern emerged. The map didn't follow geography in any simple way. It followed disease, tracing the ancient paths of humanity's deadliest pathogens across thousands of years of history, written directly into our genetics. Your blood type seems like a simple thing. O positive, A negative, B positive, AB negative, 4 basic types with 2 Rh variations, something most people only think about when donating blood or filling out a medical form. But underlying those letters is a genuinely detailed record of ancient survival, shaped by natural selection acting on human populations across an enormous span of time. The underlying ABO blood group system is genuinely ancient, with a core genetic variation responsible for it predating the split between humans and other great apes, meaning some version of this system has existed in our lineage for millions of years. For most of that time, it remained relatively stable. What changed the picture dramatically was human migration out of Africa, and the very different disease environments different populations encountered as they spread into radically different parts of the world. Populations that became geographically isolated from one another for tens of thousands of years developed distinctly different blood type frequencies, and modern genetic mapping has made it possible to actually read those differences as a kind of historical record. It's worth understanding briefly what a blood type actually is at the molecular level, since that mechanism is exactly what makes the disease connection possible in the first place. Blood types are defined by specific sugar molecules, called antigens, sitting on the surface of red blood cells. Type A blood carries the A antigen, type B carries the B antigen, type AB carries both, and type O carries neither, just the simpler underlying molecular structure both A and B antigens are built on top of. These aren't decorative or incidental features. They're the exact molecular surface that pathogens, immune cells, and other biological processes interact with directly, which is precisely why something as seemingly minor as which specific sugar sits on the outside of a blood cell can meaningfully influence how a particular parasite, bacterium, or virus interacts with the body. Blood types were only formally identified in 1900 by Austrian immunologist Karl Landsteiner, whose discovery finally explained why blood transfusions, previously a dangerous, unpredictable gamble, sometimes succeeded and sometimes proved fatal, work that earned him the Nobel Prize in 1930. It took several more decades of research after that initial discovery before scientists began seriously investigating the deeper evolutionary question of why these specific blood type variations exist and persist across human populations at such different frequencies around the world in the first place. Consider the difference between South Asia and Western Europe. Across the Indian subcontinent and much of Central Asia, blood type B reaches frequencies exceeding 30% in some populations. In Western Europe, by contrast, blood type B is comparatively rare, present in fewer than 10% of people. For a long time, this kind of disparity was simply noted without much explanation. The more complete picture that's emerged connects it to the specific disease pressures faced by pastoral societies across the Eurasian steppe, populations that lived in close, sustained contact with domesticated animals including cattle, horses, and sheep. That kind of close animal contact creates real, documented opportunities for zoonotic disease transmission, diseases that jump from animals to humans. And some researchers believe blood type B may have offered a measurable survival advantage against specific pathogens that emerged from exactly this kind of human-animal interaction. This would help explain why type B is historically rare in pre-Columbian American populations, who had far less exposure to large domesticated herd animals, while remaining comparatively common across Eurasia, where pastoral economies dominated for thousands of years. European populations show a different pattern entirely, generally split fairly evenly between type O and type A, with some Nordic populations showing somewhat elevated type A frequency. This distribution reflects a different set of historical disease pressures, ones tied specifically to the rise of dense agricultural settlement. As populations in Europe transitioned from smaller, more mobile groups into large, permanent farming settlements and eventually cities, living conditions changed dramatically. Crowding increased. Domesticated animals lived in close proximity to human settlements. Sanitation by modern standards was minimal. These conditions created ideal circumstances for infectious disease to spread efficiently through concentrated populations, generating exactly the kind of sustained selection pressure capable of reshaping a population's genetics over enough generations. Perhaps the single most striking specific pattern in all of this involves the Rh factor, particularly among the Basque people of northern Spain and southwestern France. Basque populations show Rh negative frequencies reaching into the 30s, some of the highest recorded anywhere in the world, in sharp contrast to most of the rest of Europe, and far higher still than most Asian populations, where Rh negative blood is genuinely rare, present in less than 1% of some populations. The Basque people represent one of Europe's most genetically distinct and long-isolated populations, with genetic studies indicating their ancestors diverged from surrounding European groups long before the major Indo-European migrations that reshaped much of the continent's genetic landscape, roughly 4,000 years ago. That long isolation allowed their genetic profile, including this unusually high Rh negative frequency, to develop and become concentrated in ways distinct from their geographic neighbors. What specific selective advantage, if any, Rh negative blood conferred within that isolated Basque population remains a genuinely active area of ongoing research, with several hypotheses proposed but no fully settled consensus answer. All of these patterns point toward the same underlying mechanism: infectious disease, exerting sustained, measurable selective pressure on human populations across thousands of years. This isn't an abstract or purely theoretical claim. It reflects specific, well-documented interactions between particular blood types and particular pathogens, interactions serious enough to have killed enormous numbers of people throughout human history, leaving behind populations shaped by whoever happened to carry a genetic advantage against the specific disease threatening them most. Malaria provides one of the best documented examples of this entire phenomenon. Malaria is caused by parasitic protozoa in the genus Plasmodium, and the most dangerous species, Plasmodium falciparum, has killed an almost unimaginable number of people throughout human history. Archaeological and genetic evidence suggests malaria became a significant human pathogen roughly 5,000 years ago, as the rise of agriculture and irrigation created ideal mosquito breeding conditions, combined with larger, denser human populations for the parasite to exploit. According to World Health Organization reporting, malaria caused several hundred million infections and hundreds of thousands of deaths globally in 2023 alone, with the overwhelming majority of fatalities occurring among children under 5 in sub-Saharan Africa. This is a disease actively exerting real selective pressure on human populations in the present day, not simply a historical curiosity. People with blood type O have a genuinely significant, well-documented advantage against the most severe forms of falciparum malaria. This finding has been confirmed repeatedly across systematic reviews and meta-analyses examining malaria outcomes in different populations worldwide. Individuals with type O blood consistently show less severe disease, lower rates of hospitalization, and significantly reduced risk of the most dangerous complications, including cerebral malaria. Individuals with type A, B, or AB blood show measurably higher susceptibility to severe disease by comparison, with some research suggesting AB blood type carries close to double the risk of severe malaria compared to type O. This protective effect holds up consistently across genetically and geographically diverse populations, which is exactly the kind of pattern that points toward a real, underlying biological mechanism rather than a coincidental statistical association. That mechanism is genuinely fascinating and has been the subject of considerable research. It centers on a process called rosetting, where red blood cells infected with the malaria parasite clump together and obstruct small blood vessels and vital organs, including the brain and kidneys, a primary driver of the most severe and fatal forms of malarial disease. Malaria parasites appear to bind more readily to the surface antigens present on non-O blood cells, promoting this dangerous clumping, while type O blood cells, lacking those specific surface antigens, present what researchers have described as a comparatively hostile biochemical environment for this part of the parasite's strategy. Over many thousands of generations of sustained malaria exposure, this consistent survival advantage would have compounded significantly, and it's part of why some indigenous populations in the Americas today show extraordinarily high frequencies of type O blood, in some communities reaching well above 80%, reflecting millennia of intense malarial selection pressure in the pre-Columbian era. Here's where the story takes a genuinely important turn, because type O's advantage against malaria comes bundled with a real cost in a different disease context entirely. People with type O blood are significantly more vulnerable to severe cholera. Throughout history, whenever cholera has struck, in medieval outbreaks, in colonial-era epidemics, or during the major cholera pandemics of the 19th century, individuals with type O blood have faced measurably higher mortality rates. The exact molecular mechanism behind this vulnerability remains an active area of research, but the pattern itself is well documented, and it illustrates something genuinely important about how natural selection actually operates. No single blood type is universally advantageous. Each type represents a different trade-off, offering real protection against some pathogens while creating real vulnerability to others, and the specific blood type frequencies observed in any given regional population reflect the accumulated, sometimes competing pressure of whichever diseases were historically most severe in that specific place. Cholera remains a genuine active public health threat today as well, not merely a historical one. A serious cholera outbreak in Zambia beginning in late 2023 and continuing into 2024 produced tens of thousands of confirmed and suspected cases, with a meaningful number of deaths and a notably rapid progression from symptom onset to death in fatal cases, a sobering current illustration of exactly the kind of selective pressure this disease has exerted throughout human history. In regions where cholera historically posed a greater relative threat than malaria, populations appear to have developed comparatively higher frequencies of blood type A, a pattern consistent with cholera favoring different genetic outcomes than malaria does, sometimes within relatively close geographic proximity to each other, depending on local disease, ecology, water systems, and population density. Plague represents a third major historical force shaping human blood type distribution. Across recorded history, plague pandemics, including the 6th century plague of Justinian and the 14th century Black Death, which is estimated to have killed a substantial portion of Europe's entire population, killed an enormous number of people cumulatively across the centuries. Plague, like malaria and cholera, didn't kill indiscriminately. Emerging genetic research suggests certain blood types may have conferred some survival advantage against Yersinia pestis, the bacterium responsible for plague, though the precise biological mechanism remains under active investigation and isn't yet as well understood or established as the malaria-rosetting connection. What does seem clear is that populations with significant historical plague exposure show blood type distributions distinct from populations with little or no historical plague exposure, consistent with plague functioning as yet another significant selective filter operating across human history, alongside malaria and cholera. Blood type isn't the only, or even necessarily the most dramatic example of a disease leaving this kind of measurable genetic fingerprint on human populations, and it's worth understanding it as part of a broader, well-documented pattern rather than an isolated case. A well-known parallel example involves a genetic mutation affecting a receptor called CCR5, found on the surface of certain immune cells. A specific variant of this gene, present at unusually high frequency in some European populations, provides significant natural resistance to HIV infection. And some researchers have proposed that this variant may have originally risen in frequency due to protection it offered against earlier historical epidemics in Europe, though the exact originating disease pressure remains genuinely debated among researchers. Similarly, certain genetic variants affecting red blood cell structure, including the well-documented sickle cell trait, provide real, measurable resistance to severe malaria in a single copy, while causing sickle cell disease when inherited in 2 copies, an almost exact parallel to the malaria and cholera trade-off observed with blood type O. These examples reinforce the same underlying principle blood type distribution demonstrates so clearly. Human genetics has been and continues to be actively shaped by infectious disease, with survival advantages against one pathogen frequently arriving bundled together with vulnerability to something else entirely, a genuinely consistent pattern across many different independently studied genetic traits. When researchers map these accumulated patterns globally, the result functions almost like a forensic reconstruction of human disease history. Regions where blood type O reaches its highest frequencies, sometimes 80 to 90%, point toward populations that experienced intense, sustained malarial selection pressure over thousands of years. Regions with elevated blood type A frequency often point toward populations where cholera, or comparable pathogens, exerted the more dominant selective pressure instead. The gradual increase in blood type B frequency, moving eastward across Eurasia, reflects both ancient migration patterns and long-term adaptation to disease pressures tied to pastoral economies and their associated animal-borne pathogens. The same blood type data also captures something beyond disease specifically, the actual physical history of human migration. As early human populations moved out of Africa in waves, spreading gradually across the globe over an enormous span of time, each group that settled in a new region became genetically isolated to some degree from other groups. This combination of geographic isolation and distinct local disease pressure is exactly what produced the divergent regional blood type distributions visible today. Founder effects, where a small subset of a larger population settles a new region, and purely by chance, carries a genetic profile not fully representative of the parent population, further shape these regional patterns, compounding over thousands of years of relatively isolated evolution in each new environment. Later, historical events left additional traceable fingerprints on top of these older patterns. Major Indo-European migrations reshaped genetic distributions across Europe roughly 4,000 years ago. Mongol military expansion is believed to have carried type B blood westward into regions where it had previously been rare. Silk Road trade networks gradually mixed populations and their associated genetic markers across an enormous stretch of Eurasia over many centuries. Each major wave of human movement left its own layer within the broader, cumulative pattern researchers can now trace using modern genetic analysis. What's genuinely remarkable is that despite all of this historical mixing and migration, these underlying regional patterns remain measurably visible today, particularly outside major cosmopolitan cities where extensive intermarriage across different ancestral populations has understandably blurred some of the sharper historical distinctions. In more geographically isolated regions, and at a broader continental level, the ancient signatures of disease-driven selection remain genuinely detectable to researchers studying population genetics. Modern research has continued expanding this picture well beyond malaria, cholera, and plague specifically. Studies examining hemorrhagic fever with renal syndrome, a serious viral illness found in parts of Asia, have found that blood type B appears to offer some measure of protection, while blood type AB is associated with increased susceptibility. Researchers have identified differential susceptibility patterns connected to blood type across a range of other infectious diseases as well, including dengue fever, influenza, and Lyme disease, suggesting that blood type variation across human populations has likely been shaped by a very wide range of pathogens throughout history, not simply the handful with the most thoroughly documented associations. Some preliminary research has also explored possible connections between blood type and susceptibility to certain modern conditions, though these associations generally remain less firmly established and continue to be actively investigated. It's worth being clear that malaria specifically remains a genuine ongoing concern even in contexts with access to modern medical care. Recent documented cases among military personnel deployed to malaria endemic regions, including reported cases among both American and Moroccan service members deployed to different parts of Africa in the past couple of years, illustrate that even with access to preventive medication and modern healthcare infrastructure, malaria continues to pose a real persistent infection risk in endemic regions, underscoring the parasite's continued evolutionary adaptability against modern medical interventions. This ongoing relevance has led some researchers to propose that blood type screening could eventually play a more active role in guiding malaria treatment protocols in resource-limited settings, since understanding a patient's blood type may help identify individuals at meaningfully higher risk for severe disease progression, allowing for more targeted, proactive treatment decisions where medical resources are genuinely limited. What blood type mapping ultimately reveals is a genuinely compelling picture of human adaptation in action. Natural selection, when the underlying pressure is severe and sustained enough, can meaningfully reshape a population's genetics over a relatively short span of generations, historically speaking. Malaria, cholera, and plague, operating as some of history's most consistently deadly forces, left a measurable, lasting genetic signature across the entire species, one still legible today in the simple letter printed on a blood donor card. In regions of the world where these historical diseases have been substantially controlled through modern medicine and public health infrastructure, the original selective pressure has largely disappeared, but the genetic legacy remains, carried forward by populations who no longer face the same threats their ancestors adapted to survive. In regions where these diseases remain active public health challenges today, that same genetic legacy continues to carry real, measurable clinical relevance for millions of people right now. Blood type distribution, read carefully, functions as a genuine historical record, written not in text or stone but directly into human biology, tracing the outline of ancient migrations, ancient epidemics, and the specific survival advantages that separated those who lived from those who didn't, across an almost unimaginable number of individual human lives spanning thousands of years. It's a quiet, continuously updating record, and as new pathogens continue to emerge and interact with human populations in new ways, it remains, in a very real sense, still being written today. It's worth ending on what this actually means for how we think about evolution more broadly, because it's easy to treat natural selection as something that happened only to distant ancestors, safely confined to the past. The blood type map says otherwise. Every population alive today carries the accumulated genetic residue of specific historical battles against specific pathogens, encoded not as abstract theory, but as measurable, mappable frequencies that researchers can trace back to particular diseases, particular migrations, and particular moments of crisis stretching across human history. In parts of the world where malaria, cholera, and plague have been brought under control through modern medicine and public health infrastructure, the original selective pressure has eased considerably, but the genetic legacy those diseases left behind hasn't disappeared. People everywhere continue carrying blood types shaped by battles their ancestors fought against enemies most of us will never personally encounter. And in the parts of the world where these diseases remain active, ongoing threats, that same genetic inheritance continues to carry real, measurable present-day consequences, quietly influencing outcomes for millions of people living through the same essential struggle their ancestors faced, generation after generation, for thousands of years. If uncovering the hidden history written directly into human biology is what you're here for, subscribe. Because it turns out something as simple as a blood type carries thousands of years of survival, migration, and quiet, relentless evolution inside it.